Um, Hello, I'm Arielle. So Dan and I are going to be giving you an overview of our CodeOcean pipeline and quality control system. I think you had a really good overview by Saskia just before this. So I'm going to be talking mostly about uploading and processing data. So I work in scientific computing and I manage the physiology and behavior data team, where we're really excited about gathering metadata for physiology and behavior data sessions, as well as processing them, QCing them, and packaging them into NWE. So a few of the things that we value here as we develop these systems are fast feedback, reproducibility, and transparency. So we build for both internal and external use. We want to build modular code. We want everything to be version controlled and tracked through GitHub. We like to collect extensive metadata about the sessions and the data that we gather, and that's so that we can know everything about what did the rig look like, how was the rig configured during a given session. And then we also want to develop for flexibility. So we have these modular code pieces of code that run algorithms. We want folks to be able to tune those algorithms however they need to. Um, so internally, we collect very large amounts of data, and we need to also develop tools that scale with our data and run at peak performance. And when I say that, I am really meaning that we need to give rapid feedback to the scientists and research associates who are collecting this data for any sort of data acquisition assessment. They need to be able to fail a session and retake it the next day, because here at the Institute, we acquire data 8 hours a day, 5 days a week, sometimes more than 8 hours a day. So to really be able to talk about this internal system that we've built, I want to talk about how we get the data from the rig to the cloud. So first, a user will acquire data on their machine. That machine has like maybe some sort of workflow automation that will help guide them through an experiment, while also collecting all of their metadata about their rig and their session. And then that data is saved locally on a drive. So we don't want users to have to deal with, everything that we do is in cloud. And so we don't want our users to have to deal with manually taking that data, doing any sort of annotations on that data and moving it to the cloud. We need to build all of our systems. We need to automate, all of our systems. And so when the data lands on the hard drive, we have something called the Watchdog service, which is alerted by, it's an event-based system. And then that Watchdog service will then take that data to our internally built AIND data transfer service. And what this does is it takes the data that was on the local drive, uploads it to S3. It will trigger one of our CodeOcean pipelines and store all of the metadata into our document database. And again, this is all just behind the scenes. The user doesn't really know what's happening. It just takes off once the data are acquired. And so like I told you, I also manage a team, but I also work as a software engineer, and I work mostly on our calcium imaging pipeline. So I want to dive deep into this pipeline specifically. So once that data are on cloud, we can then, this pipeline is the one that's triggered. And so I mentioned before that we build modular code, and you can see that there's lots of different things here. And each of these triangles is a Docker container, and the lines that connect them are channels in a Nextflow workflow that will basically dictate how the data move through each of these algorithms. So I also, this pipeline specifically is for multiplane data acquired on the mesoscope. And this data needs to be, or this pipeline needs to be flexible. It needs to take in interleaved TIFFs or HDF5 files, and we have a data converter that standardizes that data on input before it sends it to motion correction, where we account for any sort of brain movement during a session. We decrosstalk, which is really important for multiplane data, because during these paired imaging sessions, we'll get ghosting of cells from paired planes. I forgot to mention one thing. All of this is being done in parallel. So that's the beauty of Nextflow, is once you hit the motion correction capsule, everything is executed in all of the planes are run in a parallel compute system. We also run signal extraction to detect ROIs and extract their signals. We take those signals and account for bleach by running the delta F over F. Then we detect the given events from a signal, from each of the ROI signals. We classify those signals as either soma or dendrite by one of our models. And then we have this quality control aggregator, and I want to talk about this for a minute because, as you can see, to go from a movie of a mouse in, let's say, the visual cortex, our goal is to get to a detected event from each of the ROIs. And you see that there's a ton of steps to get there. And so there's lots of opportunities for failure, and we need to know if any of those steps, if anything happens at any of those steps that will require us to retake a given session. And so each of these capsules here, or these Docker containers, output a series of, they compute metrics, which are then passed to this aggregator, which output into this quality control JSON, which is a schema that we've defined here in scientific computing that will be loaded by our QC portal, which Dan will talk about momentarily. And then finally, we do NWB packaging for any sort of release. So I want to show you how kind of like the different steps and why we QC. So first you'll see here, this is the raw movie on the left side, and on the right is our motion corrected or registered movie. And so if our registered movie has way too much motion, we know we probably can't use it because you can't segment and extract cells confidently. The scientists need to know for the different Cree lines that they're working with, how did segmentation go? So this segmentation is actually kind of interesting. When I was talking about flexibility before, we designed this capsule, and I shouldn't say we, Johannes Friedrich did here at the Institute, where you can actually set certain parameters to either run Kaiman or Sweet2P, and within those two types of segmentation and extraction, you can then tune any of the parameters necessary to get really well-defined cells. And then we also, here's an image of the deconvolution and the detected events. So we want to be able to see that delta F over F, make sure that signal is not too noisy, that we're detecting the events that we expect. So these are kind of all of the different pieces that our users need feedback on every time they acquire a session, so they can know whether or not the data is of the highest quality. And these are different metrics that get packaged in our pipeline and sent to the quality control JSON, which will get displayed in our portal. I'll pass it off to Dan.
I'm back. I'm Dan again, software engineer here. I'm the person who was talking about Pinpoint this morning. So, by the way, if no one has mentioned it already, maybe you've gotten this sense just implicitly, but part of the idea of advertising these things is that we would like eventually for people to start hopefully adopting some of these pipelines for analyzing their tools, contributing back to them, and starting sort of converging on pipelines for eFIS and OFIS and other stuff that are shared. And the payoff, I guess, for doing that is, one, you get all the engineering power that's here in Neural Dynamics, and you also get these tools that sort of come for free if you use the pipelines and you use our metadata schema. So something to think about as we talk about this QC portal. So the idea with the QC portal is that we get a bunch of assets, the blue things, with unknown state, and we make it easy for people to tell us whether those assets are good or bad, and then query downstream in their analysis just the assets that pass their QC in sort of a standardized fashion. And there's really two pieces to that. There's a metadata schema specifically for quality control that we've developed, and then there's the portal, which allows you to do manual annotation and review of the state of the QC. So the QC portal helps you answer this question. Does this piece of data meet the requirements for my analysis? And this is what the actual portal looks like. So breaking that up into a few pieces. In the top left, there's a panel that helps you quickly see the overall state of an asset. If nobody's said this already, by the way, a single data asset to us is one acquisition. So like one session of data. That could have multiple modalities. It could just be behavior. It could be behavior and ephys and ophys and whatever else gets attached. So there's a panel that shows you the overall state. We'll go through a demo of this in a second, so you'll see this again. In the bottom left, there's a little container that shows you the different evaluations that the pipeline generated. The big panel in the bottom right is the thing that actually contains the metrics that Arielle told you about. So each metric that gets evaluated, you can look at. Some of them get automatically passed or failed, but you can also go and change that if you disagree, and then that gets pushed back into your metadata. And then the portal is a tool for people to interact with. So you can log in, you can push changes back up to the server. So before I go into the demo of the portal, let's just chat a little bit about what we mean by QC, because I think it's not quite a loaded term, but I think it's not always obvious what everyone's saying when they mean QC. So we mean quality control in terms of the data assets themselves. Like, is this session worth analyzing according to some set of rules that I've set? We distinguish that from curation, which is about filtering or reorganizing the parts of the data after processing is complete. So an entire asset might fail QC, but a single neuron might fail curation. So are my individual units good is a question about curation. Did my spike sorting pass or fail is a QC question. And quality assurance, we push back to the rig. So is your rig actually collecting valid data or not? Are channels dead? These are quality assurance questions, not quality control of the data. We've mentioned a few times that we have this metadata schema. I think Saskia briefly showed you a little bit of what it looks like. So this is just an abstract diagram. A quality control metric in our schema is a box that contains a few pieces. There's some kind of reference image. There's a name. So let's call this QC metric image contrast. There's some kind of reference image, which might be just a empty black square. There's a value that gets assessed on the reference image. So in this case, we might say that the contrast in this video is too low. And then that, through some kind of rule, hopefully quantitative in the long run, but often qualitative if a person needs to look at it and make that determination, the value gets converted into some kind of status. Any questions about the QC metadata? So if you, in your personal pipeline, can generate metrics that have these properties, you can use the portal that we've built, as long as you put your metrics into the schema that we've designed. And I would be super happy to get an email from someone in a few weeks saying, I've put my metadata in your schema and I'd like to launch your portal thing. I'd be happy to spend time helping you get that working. Okay, so let's look at the actual portal. So this is the portal website. Again, at the top is the overall state of this asset. So we are looking at a multiplane ophys asset that was collected on this date, that was run through the processing pipeline that Arielle told you about, and popped out of the pipeline at this date and time. So sometime last month, I guess, or something like that. The overall state of this asset right now is that it's pending. That means that it is awaiting manual evaluation, which we'll do a little bit of right now. Then there's a bunch of filters here, which let us filter out the different evaluations on the left side. And there's this sort of table at the top, which helps us quickly see that the thing that is pending right now is the motion correction. A human needs to come in and look at the motion correction and decide whether or not it passes QC. Okay, so we have the motion correction loaded right now. That's this evaluation. And we're looking right now at one of the motion correction evaluations. The reference image on the right, if we knew what we were doing, would tell us whether or not motion correction has passed here. I don't actually know what I'm looking for, but we could assume for a second that this looks great. It does look great. Arielle says it looks great. So when logged into the portal, the idea is that you can pick the value from a dropdown like this. We can say the quality is sufficient, and that, according to some rule, is telling us that this asset now is passing for this metric. So a person who's doing QC would come, they would look at this one, they would look at this second metric here, that passed too, and then they would go and do this for each of the different planes in this asset. And you can imagine how this might look for spike sorting. You might look at one probe at a time, look at the motion correction, the destriping, the other properties of spike sorting that were run, and make some assessment for each of those, passing or fail them. And then the idea is that when you've made all those changes, you submit them with the submit button that pushes them back into the database. And then when you go to do your analysis on these data assets, you can filter based on the QC status in the database. And that's kind of the whole idea here. So trying to standardize a little bit the process of doing quality control after running these large standardized pipelines as much as possible. This QC portal has quality control right now running from somewhere between 5 and 10 pipelines that are running on hundreds, thousands of assets by now. So this is something that scales very quickly, and that's because the schema is standardized in a very simple way and is very easily reused across all of these things. Any questions? Yeah, please.
Is it possible to release a pipeline for somebody else data? Like, for example, if I have collected my data and I want to have these QC check for my data, is it possible for other data is not from LN?
Yeah, the question is, can these pipelines be reused? Will you get the QC checks if you reuse them on data that's not from neural dynamics? So the answer is yes and no. It depends a bit on the pipeline. The hope in the long run is that all the pipelines will have sort of a set of file formatting that's expected as inputs. And for some modalities, that's pretty standard. So for example, the EFIS pipeline is pretty generic, and probably anyone could use it. Some of the other ones, like fiber photometry, I think is just one person in the building who sort of made up the rules for how the files would look. So that would be much harder. Yes or no? Is that a yes? Anyway, so we're at different points on this sort of spectrum of reusability depending on modality. But the long-term goal is that the engineering work here will benefit the community as much as possible. And that's also the point of having these workshops and talking about this stuff. To the second part though, do you get the QC for free? Yes. If you run the pipeline, it generates the quality control metadata in the schema. It generates all the images that need to get attached as references. Everything's there. There's a little bit of overhead that you'd have to deal with after that. So we of course have our S3 buckets that are all set up nicely and that work with CodeOcean, and the portal pulls from them directly. So there'd be a little bit of work to convert the system to use sort of your lab infrastructure. But the goal is for that to be easy in the long run. Yeah.
Are these only in Python? Is it adaptable into other languages?
The pipelines are written in Python.
Okay.
Converting them to other languages, well, I think the hope is that you don't convert them because then you're contributing back and everyone's benefiting from things that are getting added. Yeah.
Okay.
What are you trying to convert them to?
Something like Julia or MATLAB for like analysis after the fact. Like, would it be easier to have an output be into one of those language formats?
So I'm gonna probably repeat something that either David or Saskia said earlier, but we really think of the processing pipelines as being sort of the standard set of stuff that everyone wants to have done up to packaging into an NWB file. And then after that, it's sort of a free-for-all. And partly that's because we know that to do fast iteration and have experiments run quickly, there's only a small set of things that you can do on all assets, right? And that's really what we're pushing up to this sort of processing, or what other people might call pre-processing stage. Yeah. So you can still do whatever you want after you get your NWB file.
Your CodeOcean runs Docker, within which you can run many different languages. So, for instance, you can have MATLAB in there, but you have to be familiar with CodeOcean.
Yeah, another question.
We've already said this, but could I upload histology data, not just like EFIS or like calcium imaging? Does it have to be like a time series?
For what exactly? For the pipelines or for the QC portal?
For the QC portal.
So the QC portal actually has no knowledge of the data at all. It's pulling only from the metadata schema, which is a big JSON file. I don't think I have time to show it exactly, but we could quickly look at it afterwards. So there's a JSON file basically that has these information about the metrics. It's really just this information. And all the portal is doing is reshaping this into the view that you're seeing on the website. Right? You can see again, there's a value field here with some options. There's the status and the reference image on the right. And that's really it. So it's just a folder with a bunch of images or PNGs or PDFs or other stuff. And then this little bit of metadata on top of that. Yeah, so the answer is absolutely. You could generate that metadata schema for anything. It doesn't even have to be neuroscience data, and then use the portal to view it. Okay. So the last thing I was going to say is one of the things that we're working on is this curation step. So folks who are familiar with Phi or who saw it earlier today, a lot of people want to touch their spike sorting results and really check that the waveforms look good and split units and remove units that look bad. And we think of that as this curation step. So it's not really about the data asset itself. It's about the parts of the data asset. So one of the things we're doing is building into the QC portal apps that allow you to do that curation step in this sort of shared space. So I don't think you can see it very well, but this blue thing on the right here is a sort of simplified version of Phi called Sorting View. It runs on the web and allows you to do a little bit of what Phi does live. And we're working on building a sort of more performant version of this that's just a replacement for Phi and runs in a browser, which we'll probably have in 6 months or a year or something. And we have a bunch of these hopefully coming in the future, not just for spike sorting, but for other data modalities where you want to do this kind of curation. And I think that's it. You're welcome to play with the QC portal. It may crash if too many people try to access it at the same time, but if you wait a few minutes, it'll always come back eventually. So you're welcome to go play with it now. And this is part of the AIM data schema sort of ecosystem we're trying to build. Tools where if you adopt our metadata schema, you get the tool for free. So this is the first time you'll hear that, but over the next few years, if you ever see me give another talk, I'll keep pushing that line. Over. Thanks. Happy to take questions, and Arielle's here too to take questions about any of the pipeline stuff.